I'm Natalie Strickland. I'm the Global Head of Translational Science at Synexa Life Sciences. The process that we use for our translational services offering is very customizable and adaptable. We pride ourselves on being flexible and working with our customers to understand their needs. That starts off firstly with consultation with the customers and understanding the challenges that they're facing, the questions that they're asking and that they require answers to. This is typically done with a very experienced scientific team, talking scientist to scientist to understand how we can support and offer them a service that's ultimately going to be of value to them. And we then start working on the protocol. So this is really important, and one of the critical aspects of the translational service at Synexa is that we prepare protocols which are then submitted to a local ethics committee or IRB for approval prior to collecting samples from human patients. So we work with the customer to design the protocol. We have a lot of experience with this, so we can offer guidance. We understand what the local ethics committees require from these protocols. Once that is complete, we move into the experimental phase or the assay phase. This is when the samples are collected from the patient groups that we're targeting. We do this in collaboration with our local clinician network that we've established over 20 years now. We have very good relationships with local clinical sites, key opinion leaders in their respective fields, and we work with them to collect the samples that are relevant for that particular study. Once we have the samples, we then can apply the various analytical technologies that we've discussed up front with the customers to these samples to ensure that we're generating the data that's going to be useful and informative for them. Once we've collected all the data from the samples, we move into a data analytical phase. So this is when we will apply various technologies to look at the data, really driven by the nature of the study and how it was designed. So we have a number of approaches for either looking at integration of multiple data sets from different technologies, or just providing deeper insights from highly multiplex technologies such as nanostring or Olink. Once the data has been generated and reviewed, we then share that with the customers. We have a number of different routes to do that. We typically provide interim reporting, which allows everyone to stay on the same page and not have to wait for end of study for data to be shared with them. This can really help in the design of future clinical programs because you get an early insight into, for instance, potential biomarker strategy that's coming out of the data we generate, and it can help you design your clinical program. So it's a very iterative, collaborative way that we work, and it is to a large extent driven by the specific study or customer needs, but there are specific sort of touch points throughout that we follow from a standard perspective. At Synexa, one of our key values is improving the quality of human health. And we really pride ourselves on playing a part in that entire value chain, providing help to our customers who are developing the therapies that will ultimately treat the patients with these diseases and ensuring the success of their clinical trials.